17th, 7 p.m. We have not one, not two, but three of our graduate students who will be speaking about their research. We've got Willow, Stephanie Petrovic. Oh no. Well, it's definitely live streaming because I just watched myself on YouTube. Um, so again, my name is Ariel Firebaugh. I'm the Director of Scientific Engagement here at Blandy. I'm so excited to welcome our two grad student speakers tonight, Ethan Scutches and Mia Murray, who will be sharing about their research. Both Ethan and Mia study native trees. We didn't plan it that way. It just happened. So we've got two tree talks this evening. Imagine that at an arboretum. Now, if you didn't know, in addition to being home to the State Arboretum of Virginia, Blandy Experimental Farm is also a University of Virginia field research station as part of the Department of Environmental Sciences. So Blandy is home to a community of 20 to 30 students and scientists every year. And actually, we've got a big milestone coming up. So the first Blandy graduate students arrived in 1928. So we're coming up on 100 years of students who have called Blandy home, use this as a home base for their ecological and environmental science research. And look at all of these cute Blandy scientists over the years. So it's Blandy Centennial. And we're so excited about the centennial that we are stretching the celebrations out over 2 years. We're kicking it off this year, and we're just going to celebrate as long as we can. And we're ended up here with two of our current Blandy farmers who will be our speakers tonight, Mia Murray and Ethan Scutches. I'm really excited to learn about their work, and I hope you all are too. So with that, I think I'm going to pass things over to the first of our speakers here.
Okay, I'm going to share my screen now.
Mia is one of our repeat Blandy farmers. She was an undergraduate researcher, and she came back.
Okay, well, thank you so much, Ariel. Thank you everyone for being here. Like Ariel said, my name is Mia, and I'm currently working with Dr. Ty Rolston at Blandy for my master's work on butternut trees in Virginia. So to begin my talk, I just want to give a framework to the conservation of a species and what that looks like and how we're trying to approach this problem with butternuts here in Virginia. And so first it involves recognizing the downward trend in a species, and that's done through inventorying and research and monitoring of a species at local, regional, and national scales. And then once those causes are recognized, or once that downward trend is recognized, we look for why that population decline is happening, whether it's for environmental reasons, disease, humans, whatever is happening. And from there, we look to identify how we can prevent those trends from happening and recognize how we can support those populations, either through captive breeding, land management, environmental regulation. And then from there, work to enact actions to continue to support and promote the upward trend of those populations at the policy and institutional level. And then from there, we hope to reverse those trends. And so before I jump into butternuts, I just want to give this and put this in context of another example of trees that is familiar to Blandy, which is the American chestnuts. And the American chestnuts are a tree that was very prominent in the American landscape. And then in the early 20th century, there was a rapid decline in the species, and they attributed that decline to the chestnut blight, which is a very fast spreading disease that wiped out most of the population very quickly. And so in order to, because of the rapid decline which was just happening and the fact that chestnuts were pretty heavily logged, there wasn't really opportunity to find resistance in the wild. And so rather than be able to find resistance, to be able to further those restoration efforts, they had to create resistance, which was breeding with the Chinese chestnut, which is resistant to the Chinese blight. So they're combining the blight resistance in the Chinese chestnut and the hardwood type in the American chestnut to promote the restoration and the economic viability of the species. And so now let's talk about some butternuts. Oh, and after this, before butternuts. And so in order to work with the chestnuts, they wanted to mass produce, and these orchards and plantings are happening throughout the state, including at the Virginia State Arboretum, or Blandy, with American Chestnut Foundation. So now let's get into butternuts. Okay, so butternuts belong to the Juglans family, or the walnut family. They can grow to be 60 to 70 feet tall, and they have some distinctive morphological features that you may be familiar with if you've seen them around. First is this gray colored bark with this flattened, almost diamond-like pattern. They have compound leaves with a terminal leaflet, and the leaflets follow are similar in size throughout the entire leaf. And then they also have, and which is arguably one of their most distinctive features, is these fruits that are elongated, and they typically are in bunches of 3 to 5 on the tree. And so why should we care about butternuts? They've been used by humans and animals for centuries. The nuts are really high in calories and protein, so they're super important for overwintering animals, especially in the northern region where there are no black walnuts, so they provide that resource there. They also are an important larval host for species like the Luna moth. There are many indigenous uses of butternuts in this region. We are in southwestern Virginia. We are at the northern part of the Cherokee region, and the Cherokee used the butternuts, or the butternuts were featured as one of the 46 foods served at traditional feasts. In addition to indigenous uses, they've been used, the husks of the fruits were used for dye for Confederate uniforms. And I think this is really interesting because it kind of gives us a baseline of what the butternuts were at one point in history. If they were common enough to be one of the main sources of dye for Confederate uniforms, then at one point they must have been super prominent on the landscape. And then now they're still used for wood carvings and dyes and stains, and people still consume and use sap, collect sap to make a sweet syrup, kind of like maple syrup from maples. And so within the United States, butternuts are found in the eastern part from the Great Lakes up into Maine and then down into the southeast. And then within Virginia, they're native to these counties highlighted in green, which are the western and central parts of the state. And while you can still find them in this pink region, the coastal plains, they are native to these green counties here. And so within Virginia and across the region, their actual habitat requirements is high light. So they require a lot of light in their early stages to promote the growth and survival of seedlings. So you can find them primarily along riparian areas and roadsides, but this also looks like open fields, gaps in the forest, and open ridges. And so we know how butternuts have been used and why they're important and where we can find them, but why are we trying to protect them? And there's 3 main reasons why the butternuts have faced serious decline over the past several decades. The first is the Butternut Canker Disease. This is caused by a fungus that is not native to the United States, but we're not sure how it got here or where it was introduced. But the Butternut Canker Disease essentially causes these canker sores on the trunk, and so the stem starts girdling, and nutrients are prevented from reaching the leaves, and so it eventually kills the tree over time. There's no known cure for the butternuts and for the butternut canker disease, and we do not we have not found full resistance for butternut canker disease among pure butternuts yet. Another main threat to butternuts is a loss of suitable habitat. So with the destruction of riparian areas due to dam construction and the reduction of forest disturbances like fire that provide those open canopy and highlight situations, the ability for butternuts to have ample light for early stage growth and survival, coupling with the butternut canker disease, causing mass die-off, is two main contributors to the decline of butternuts in the US. And then this third threat to the butternuts is a little bit more tricky and can be considered a threat or a potential opportunity for resistance, depending on how you look at it. And this third threat is the hybridization with the Japanese walnut. The Japanese walnut was introduced to the US in the 19th century for nut production. And since then, it kind of escaped its realms of agricultural use and started hybridizing naturally with the butternuts in the wild. In some areas, the hybrids outnumber or are more dominant than pure butternuts in the landscape. But interestingly enough, the Japanese walnut is resistant to butternut canker disease. So what do we know? We know that there's no complete resistance found in native butternuts. We know that hybrids have more resistance to this butternut canker disease. Heritability, or in other words, the genes or traits that are passed from parents to offspring, so in this case, resistance to the disease, is weak, but it's there. And we also know that there are lineages of pure native butternuts with resistance, but those are rare. And this is really important in the context of Virginia because Virginia has been very poorly studied. And so this image is from a study. It was published in 2017, but it took place earlier where they were looking at butternut presence and abundance between two time periods, first in like the late 80s and early 90s, and then again in the 2010s. And we can see that all these time periods and all these comparisons took place throughout a lot of the butternut region, but Virginia was overlooked in that study. So now putting it back in the framework of the conservation that we talked about earlier. So we know that the butternuts are declining, and this varies from 50 to 90% depending on what state you're in within the region. We know that the causes of decline are because of the disease, because of loss of suitable habitat, and the hybridization or the replacement of pure butternuts by hybrids in the wild. And so moving forward, we know that the next step to restoring and to mitigate further loss is to breed disease-resistant trees for reforestation. But how do we get resistance? And so my project aims to get a baseline knowledge of what's happening with butternuts here in Virginia and be able to lay the foundation for finding resistance here by locating as many trees as we can, by genetically sequencing and assessing the health and habitat of those trees that we find, and then being able to collect seeds for future propagation efforts based off of the genetic and health assessments that we find. And so my research questions ask, where do butternut trees remain within their native range in Virginia? What is the rate of butternut canker disease in the butternut's native range within Virginia? And then how does the health of butternuts vary across genotypes, so pure or hybrids? And so what have we done so far? So over the past 3 years, we have sampled many trees, but we've been able to actually send leaves to be genetically sequenced from 68 trees. And from these 68, some had to be discarded because they were misidentified. One was a hybrid, but the majority were those pure were pure butternuts. In addition to the genetic sequencing from the leaves, we also scored various trees for canker presence. And the canker presence in the wild range from little to no infection to severe and heavily infected trees that had nearly total canopy loss. So there's a wide range of cases and scenarios that we've seen, but we know there's way more to uncover. So in trying to find as many trees as possible, last year we did a first major preliminary field campaign to find trees, and that took us from Blandy to the southwestern corner of Virginia on the border of Tennessee. And we were basing these stops off of iNaturalist sightings of butternuts. So we were assuming that some of them probably weren't going to be accurate, but we were hoping that a lot of them were. And without verifying these trees, we came to and spent a lot of time driving to sites that actually had no butternuts. So we want to create a more efficient use of data collection and location finding, but we're also limited by the knowledge of where we know butternuts are just from where we encounter them. So what needs to happen to find these trees? So we are working to find trees, to collect leaves, and to collect that data for the health and the size and the habitat. We will send those leaves off to be genetically sequenced. Once we genetically sequence those trees, trees that are pure hybrid, that are pure butternuts and that are large and relatively healthy will then have their fruits and seeds collected from them. And then those seeds will be planted and begin the process of the next phase of research that includes propagating trees for future resistance testing. So the first step is locating trees. And like I alluded to earlier, we've done some of this, but there's a lot more trees that we need to find. But this huge range is nearly impossible to find with 1 to 2 people driving around in the Blandy van looking for trees. And this is why we need help. And so there was a post this past summer, 2 summers ago, that Blandy put out about butternut trees, and that received a lot of excitement from people telling us where they knew of trees and talking about their connection with the tree. And so leveraging people's local knowledge, we're trying to build a citizen science network that will include people helping locate trees for us and even collecting data and sending us those samples. So I've created 3 roles that people here in Virginia can be a part of that process of collecting and finding trees for us. And you may fall into one or multiple of those roles. So over the next few slides, I'll talk about what that looks like. So first is a sampler role. So this is someone who has a tree on their property or has a friend or a neighbor with a tree on their property, and they think they have a butternut. So first they confirm the butternut tree with us by sending us photos to an email, and that turnaround time will be within a few days of us determining it's a butternut just by looking at the photos. And we want those photos to be of the bark, of the leaves, of the fruits. Obviously right now it will be harder to do that, but if you have prior photos, that's great as well. And so once we confirm that it's a butternut, you'll receive a package in the mail from us with protocols and guides, as well as a prepaid shipping label to send the samples back to us. And then you will take the pictures for us and send those back to us via email, and then you will mail us the leaves, and then we'll send those leaves off to be genetically sequenced. Next is the sharer role. So this is semi-similar to the previous role, but you are not collecting the data directly and mailing it back to us. We're assisting in that process because maybe the tree is too tall so you can't get the leaf safely, or maybe you've had a recent injury so it's not feasible for you to do the data collection process yourself. So similar to last time, you'll confirm through email with photos that it's a butternut tree. And then if it is a butternut tree, we will come and do the data collection process ourselves, or you're more than welcome to help us with that process. So you can be as involved or not involved with that process in this role. And then we'll take those samples back to our lab and send those off to be genetically sequenced. And then for the final role, the surveyor role, if you find yourself hiking in Shenandoah or in George Washington National Forest or any sort of state national park within the state bounds, you may come across a butternut tree, but because it's on state or federal property, you're not allowed to do that data collection yourself. So if you, now that you know this project, if you come across trees that potentially could be a butternut, we want you to share those pictures with us, share those coordinates, and then we'll confirm. And then if we don't already have the permits for those areas, we'll go back and collect the butternuts ourselves, and then send the leaves, collect the leaves ourselves, and then send those off to be genetically sequenced. And so just to reiterate, the sampler is someone who's directly doing the data collection for us and mailing it back to us. The share is someone who's granting us that permission to come and either allow us to do the data collection ourselves or assist us with that process. And then someone who's surveying and happens to find butternuts and sends us that location data so that we can go back and collect that ourselves. And so once those leaves are genetically sequenced, we'll be able to cross-reference the pure butternuts with those field assessments that we did, looking at the size and the health of those trees. And the trees that are large and relatively healthy will then be returned to collect seeds. So this part is really important for those first two roles that I talked about, the sharer and the sampler, because those people who have direct access to their trees will then continue to monitor those fruits or monitor that fruit production. And if you end up having a tree that fits into this category, you'll let us know when those fruits are ready. And then typically that would be in like mid to late August, and then we'll come back and collect those seeds. And we're collecting those seeds because we'll begin the propagation phase of the project. So we'll come and collect the seeds and then bring those back to Blandy and then start the next phase of the process that looks at propagation and then transplanting those out and beginning to do resistant experiments to test susceptibility, resistance, and all of those things. And so what are the outcomes? What do we want to get out of this data collection? We want to gain insight on butternuts on the southern end of their range. We want to create a comprehensive assessment on remaining health, genotypes, and locations of butternuts here in Virginia. We want to begin to identify potentially resistant lineages to continue to, or to pursue that resistant experimentation on, with the ultimate goal of restoring more resistant butternuts to our forests. And so being a part of this project, there are deliverables and skills that can be gained. So learn about tree and disease identification, gain hands-on data collection experience, and learn about scientific protocols. One of my most important outcomes is that this data is available to everybody who helped us collect it and doesn't just remain within the walls of academia. So creating a final report so that people can see not only the data that they contributed to, but what their neighbors and what people around throughout the network contributed to. This is also an opportunity to work and learn and meet people in federal agencies, environmental nonprofits, and other researchers, as well as begin to form this larger scientific network throughout the state that serves as a continued place for knowledge exchange. And so the timeline is right now, we are in the phase of contacting and recruiting people to help us with this project. So March and April and into May, finding as many people and finding as many trees as possible. April, hoping to have a training in person at a central location to walk through protocols. And then if you're in that sampler role, you'll get those materials in May. In June, we'll collect leaves in hopes that all of those leaves are collected by the end of the month so that we can send them off and take that genetic sequencing turnaround takes 4 to 6 weeks. So those will be sequenced throughout July. And then once we have those results, the candidates, so pure, large, relatively healthy butternuts will be returned to in August to collect seeds with an ambitious goal of having the report in everyone's hands in the mid to late fall. And so to bring it back to round out the presentation, the tale of two trees, the chestnut and the butternut. So both of these trees, the main goal is maintaining resistance, getting resistance to restore more resistant trees to the wild to prevent another mass die-off. And in the case of the chestnut, because it was so heavily logged, it wasn't feasible, or it was going to be much harder to find that natural resistance. Therefore, they had to create it with the breeding with the Chinese chestnut. But in the case of the butternut, we still have time to find that natural resistance. And what this project is built on is years of research from people across the range. So it's not a matter of knowing what to do. We know what to do. It's a matter of how quickly we act and how well we do so. So this project is not possible without the beginning work started by Purdue and the Forest Service and the Morton Arboretum, and just excited to have Blandy be a part of this larger project. And thank you to the Virginia Master Naturalist and Botanical Garden of the Piedmont and the other environmental nonprofits that have already supported us. And thank you to everyone who is already reached out to us with locations, and hopes that maybe some people on this meeting potentially know of butternut locations and we can form new partnerships. So thank you, and I'll be happy to take any questions.
Thanks so much, Mia. So if this is a good time, if you've got questions that are just burning a hole in your little brain right now, feel free to type those in the webinar chat. But as you're thinking and typing, if you are the kind of person who has always wanted to participate in a real scientific study, this is your chance. Mia needs you to be on the lookout for butternuts. So next time you go out on a hike, next time you're walking around in your backyard, take a look at the trees, see if you have any butternuts back there. Okay, we've got some stuff coming in through the chat. Maggie says, great talk. She's excited by the community science aspect of your project. Very cool. Couldn't agree more. And Scott says, nice job, Mia. Where do the samples go for genetic testing?
That is a great question. We are still working out where that exact location will be. The genetic results that we did for the past few years were sent to Purdue University. Purdue is really the biggest player in the butternut game right now, really the main player in the butternut game. So that's where we sent it in the past. But depending on the turnaround time and the other projects they have going on, that may be a little bit different, but we're hoping to continue that with them.
That's great. And Isabella says you should use TreeSnap for citizen science. Are there any apps that you would recommend, Mia, for people who want to learn how to identify butternuts?
Yes, kind of. Originally we really wanted to use TreeSnap. And there are some butternuts located on there, but it's not as updated as something like iNaturalist, because it's not as well known as iNaturalist, and I think the interface is a little bit more difficult, and it takes longer to get verified because TreeSnap is maintained by like foresters and that stuff. But we're hoping that through this, one of the outcomes of this project is that we can continue to support the like natural history online databases that are accessible to people. So yes, I know about TreeSnap. TreeSnap and iNaturalist are great apps to use, and we want to continue to support that. So we have used some TreeSnap, and that's what some of the previous stuff has been done. But moving forward to cover the range that we want to do, we're trying to contact people outside of TreeSnap.
And when in doubt, they can always email pictures of trees that may or may not be butternuts to UVA butternuts at virginia.edu. Well, thanks so much, Mia. I think we're going to pass the baton over to Ethan. But if another butternut question occurs to you out in the audience, feel free to put those in the chat. We can circle back to those at the end.
Oh, all right. Let me figure out this screen sharing situation. Oh, I figured that out quite quickly.
I see a tupelo tree.
Okay, amazing. All right. Um, I guess I'll get started. So today I'm going to be talking a little bit about my research. Well, I'm Ethan. I'm a PhD student in environmental science. I'm working with Dr. Megan Blumstein. And today I'll be talking a little bit about my research and the processes of restoring resiliency and my work with the tupelo tree. So to get started, very like baseline information that sets the scene for a lot of work that people do in this field, including me. Our climate is changing and our forests are changing in response to this, and we have to be ready for it. So we know, hopefully, fact, the climate is changing. We can see this over time. And just to kind of like drive this home a little bit, I'm going to use the USDA hardiness zones as a reference point. So these zones are set forth by the USDA, federal organization, and they're based on average annual minimum winter temperatures, typically, well, now from 1990 to 2020. So they don't include these freak days like we had today, where yesterday it was 80 degrees and today it was 30, but they're based on averages. And so these are displayed across the country as 10-degree Fahrenheit zones, with zone 1 being the coldest and zone 13 being the warmest. And so you can see on this map of the country, you have your colder areas represented in like this light purple bluish, and then your very hot areas are red orange. And so we typically function on a gradient from north being colder and south being warmer. And these hardiness zones are generally used and like serve the purpose as general guides for growing perennial plants across specific regions across the country. And so if you're a gardener, you'll probably refer to this a lot in terms of like, what can I grow in my garden year round, both whether that be a vegetable or a native plant. So thinking about Blandy, which is in Boyce, Virginia. This is where we were looking in 2012. We were in USDA Zone 6B. These are just all numbers. They have associations with what plants you can grow to them, but we're just going to look at the numbers for the moment. We have 6B, where our little heart is, State Arboretum. We love her. But then you have some 5B and 5A as you go farther west towards West Virginia, and then Washington, D.C. right there is 7B. And then in 2023, the USDA updated their hardiness zones for the first time since 2012. And it was a big kind of wake up call to a lot of people working in the horticultural plant botanical field at large. And so we saw a shift in Boyce, Virginia, where we went from 6B to now zone 7A. Washington, D.C. is zone 8A. And then out west, where we were kind of having those more fives, we're really kind of seeing a bit of an erasure of the 5 range there. And so again, these are just numbers. They pertain to plants, but there's this thought of like, we're seeing this warm up, and we're seeing it specifically in urban centers and then kind of radiating out. And so we saw that happen over the span of 11 years. We shifted 1 zone. So in the next 11 years, at the rate of which we are warming and the earth is warming, what zone are we going to be in next? And so all of this to say, now it could be argued that Washington, D.C. is suitable for lemon trees. And I have this really great graphic that I made with a little bit of clip art that maybe we can imagine the beautiful cherry blossoms that line D.C. are now lemon trees. And that's a crazy thought. The climate of DC is really now more representative or very comparable to that of Atlanta in terms of what you can grow year round there outside of like these freak freeze events. And so not only is our climate changing, but it's changing very fast, and it's affecting how and where plants can grow. And so my research focuses a lot on this general question of, can our natural environments keep up? Thinking about specifically forests and trees. And so if they can keep up, that's awesome. I will back away. I don't need the PhD. I'm fine. But what if they can't? And thankfully for my funding, but unfortunately for our environment, they're having a little bit of trouble. So trees can respond to environmental change in a variety of ways. They can migrate, so that's been seen throughout history. This is a multi-generational and long-term movement of trees in response to climate. And so an example that we're seeing of this more often now is trees migrating up mountains. So typically with a mountain, you would have a tree line where once you hit that tree line, you're really dealing with rock or small shrubs or a grass layer. And so this photo below is a photo from the Appalachian Mountain Club, and really looking at this area in the kind of upper left corner. This photo on the right is from 1978, and then this photo on the left is from 2014. So you're seeing a lot more green up there, a lot more expansion of the forest. So it's getting warmer. If I'm a tree that likes to be mid-range in the mountain and now it's getting too hot for me, I'm going to crawl a little bit farther up and up and up and up. And so they can do that. They can migrate. They can also adapt. And so an adaptation in this context is a very long-term evolutionary response to a specific environmental stress or pressure. A great example of this is honey locust trees. And so these are a tree that's fairly common around Virginia and up and down the East Coast. They are an example of adaptation with them is that they can grow thorns, very large thorns like those in this picture. And those are specifically to defend them against megafauna. So it's believed that they were being browsed on by like woolly mammoths way back when that was a thing. And so to combat that, they said, I'm now going to grow thorns to say, screw you, do not eat me. And so, in a similar sense, the trees can also acclimate. And so the difference between adaptation and acclimation is that adaptation is a long-term evolutionary response. So this occurs over generations, whereas acclimation is a short-term response, something that occurs within a single tree's lifetime. And so an example of this is with the American holly tree. And this photo I actually took last week. It was cool to see in person because I've always heard about it, but holly trees they typically have these very prickly, pointy leaves that are really painful to walk against or engage with if you are doing any sort of work near them. And that is a defense mechanism against herbivory. So typically they will put out these leaves in response to a deer eating them. And they kind of in a similar sense of locusts, they'll say, screw you, do not eat me. I'm going to have thorns now. But higher up in the canopy where they are not being predated, they will have more elliptical round leaves with no points. And it's better for photosynthesis. And it takes less energy to have a perfectly round leaf than it does to throw out these little dagger thorns. And so these are ways in which trees can really adapt and respond to this, or broadly respond to this change. But what if they can't? So they die. Um, Hate to see it happen. It's the sad truth. One of the main kind of like a lot of times when we think about like evolution and stuff like that, it's kind of like evolve, adapt, die. Those are your options. And so extinction is a very real threat, less so with tupelos, which I'll talk about, but it's something that we're seeing a lot in response to a warming climate. You see this both on the local and global level. In the lower left-hand corner here, I have a photo of bunchberry, which is in the dogwood family, and it's state endangered in Virginia, predominantly because where you're going to find it is up in the Appalachian Mountains. But as our climate is warming and you see species moving themselves farther up the mountain, those species that already call the tip tops of those mountains home, they have nowhere to go. And so when it gets too warm for them, they disappear. And so you can have extinction, both on the local scale. Let me be so clear, bunchberry is not threatened globally. It's very happy where it currently predominantly resides in northern New York and Canada, but Virginia, it's a bit less happy. You also have invasive species dominance. So if you've driven anywhere in the southeastern United States, you have probably seen infamous highways of kudzu. These can really proliferate under climate conditions. When you have these warming climates, you have stressed out trees. They are less apt to be able to shade out, or they have 8,000 other things going on that stresses them out. And so a vine can really easily take place, and then they form a monoculture, and then your forest looks like a beautiful English garden, but ecologically, it's not really functioning. You can also have forest dieback, and then you can have increased pest susceptibility. So like Mia was talking about with Butternut Canker Disease, that spread is exacerbated by change when you have all these other factors that are already stressing out a tree, throw on a pest or pathogen, it's a pretty tough life to live. And so I'm really interested in like, I just bogged you down with like, oh no, they're dying. But how can we help? And so I'm really interested in kind of how can we facilitate this, any sort of adaptation, migration, acclimation in order to not have extinction. And so restoration ecology, very broadly speaking, is a field of practice within ecology at large, but it focuses on restoring function to a system and aiding in the recovery of a system. It's a bit of a misnomer because when you think of restoration, you think of going back in time with a changing climate. It's really imperative that we use history as a baseline, but we're not really at like a point in time now where we can use history as a goal. We've changed so much. There's so much happening all the time. We're constantly changing. It's pretty hard to rewind the clock. And so, in examples of restoration ecology, it's not some like coveted thing that you can't do. And it's as simple as cutting a vine, freeing a tree from a vine, engaging in any sort of tree planting, invasive species removal, putting in a pollinator garden, all of that sort of stuff. But rather than thinking about how systems once were within restoration ecology, we're really starting to think now about what they will be going forward. And so thinking about the forests of the future, and specifically what I think about a lot in my line of work is forest assisted migration. And so forest assisted migration is a climate adaptation strategy which involves the intentional movement of trees to new locations. This diagram on the right shows you two kind of scenarios that fall under the broad lens of assisted migration. So you can do population migration where, you have, I'm going to use tupelo as an example, but you have a lot of tupelo growing in Virginia and you have it in Texas, and you just want to kind of mismatch. You want to get some more genetic diversity in there so you can pluck some Virginias and pluck some Texas and then bring them back and forth within one another and to get some genetic diversity up. You can also do assisted range expansion. And so this is thinking, okay, we know the native range of tupelo. We know the native range of all these plants. Let's see if we can push the boundary a little bit. Let's move them outside and see what happens now. And so this whole concept of forest assisted migration is that it's anticipatory. We are moving plants to areas better suited for a predicted climate. We can't really ever be 100% certain. But the way that I look at it is that I would have rather tried something than kind of sit stagnant and watch these systems disappear. And so I like to think about restoration ecology with the inclusion of forest assisted migration as restoring resiliency. And so we're preparing forests and ecosystems for transitions in climate. And so when I think about forest assisted migration, which I'll just refer to as FAM from now on, I lean more on the assisted range expansion side. And so we know that like forest assisted FAM, if you will, is a kind of evolving concept. And it really came out onto the scene in the early 2010s. And there was a lot of controversy about it. There still is some controversy about it, about whether or not we can be moving plants around, but also where should we be moving trees from? What are the genetic implications of humans migrating trees? And how are specific species related across their populations or even within sites when we want to pick a tree? If we're going to move tupelo, do we want to move that from trees in Alabama? Do we want to move that from trees in Texas? Where in this range should we be picking from and why? And then what are effective means of migration? Should we be relying on birds and animals that do this on the daily for us? Or do we really need to step in? And is it as simple as planting a tree in front of your yard? Is it a really big reforestation project? Or is it something like planting a tree in a city? And so now I'm going to talk to you a little bit about tupelo. Tupelo has a lot of names, so I am referring to it in this presentation as tupelo. It's black tupelo, black gum, sour gum. All of these can be used to refer to the tree of Nyssa sylvatica. And so it's in the same family as flowering dogwood. It's a deciduous tree species, so it loses leaves in the fall. And it's found from the understory to the canopy level of the forest, which is pretty funky. It has this chunky bark on the right. I think it's a stunner. I love it. And one of the really most important characteristics that I like to hone in on about this tree is that it's facultative. So it can grow in a variety of environments, whether that be dry uphill sites or wet river adjacent swampy sites. And so some other examples of trees like this are red maple, sweet gum, and eastern hemlock. And I refer to these trees as like sleeper agents a little bit because they're often looked over in this, specifically tupelo. I think tupelo deserves a bit more time in the light. And it's starting to gain some more use as an urban street tree. And so the range of tupelo, I have on the bottom left here a photo from my naturalist, and then also this map on the right that I'll explain. So it's found across eastern North America. The range really spans from eastern Texas to coastal New England and up into the Hudson Valley. And we dip a little bit into Florida down there as well. It comes up the Midwest as well, but it's typically east of the Mississippi. There's like one disjunct population to the west of it. It's historically like where we are in Virginia is really the most concentrated area, or a very concentrated area for the population. I see tupelo all the time up here. Before I was in Virginia, I was living in the Hudson Valley in New York. I was elated to come across a tupelo there because it was so uncommon. And so this photo on the right just shows that while we have a historical range of tupelo, it's moving. And so the gray here shows the historical kind of understood native range of tupelo, and these green splotches are where we're seeing some migration. And so this is just with the Forest Service data. So they really rely on occurrence data, but this map here on the left is more citizen science data. All those orange points are there's a tupelo there. And so we know that it is kind of extending past its range, and we're seeing that movement north. So how is it doing this? Probably by birds. Fruits of tupelo are enjoyed by a plethora of birds. On the right side here, we have a pileated woodpecker eating some tupelo. Then we have an American robin on the bottom right, and then a cedar waxwing in the middle. And this is a photo here just of the fruit. And so they're moved around by birds, but also just plants in general are moved around constantly by humans. They're planted as street trees in urban centers. They're sold in garden centers, all of this sort of stuff. So there's countless ways. It's really hard to track exactly how trees are moving and plants are moving, but we know that they are. And the question that I have here is, which tupelos are we moving around? So my research is a little bit of genetics. I'm going to talk a little bit less about that today, and more about the more field work that I'm doing at the moment, but we'll get into it. So I'm really interested in how tupelo are related across their range in the wild. So thinking of making like a family tree for them. How are they related within a site? And then specifically going back to tupelo being facultative and really versatile and where they can live, I'm really interested in how they are related within wet and dry populations. So even though these trees both grow in Virginia, if one is growing right next to a riverbank and one is growing really uphill in a really dry area, do those trees have different genes that enable them to thrive in those specific locations? Do they have different genes that will enable them to respond to different climate events better? With this whole change in climate thing, we're seeing more intense drought, we're seeing more intense heat waves, we're seeing more intense fluctuations in just the pattern of weather. What trees are going to be more adapted to that? And is that encoded based on where you're growing? And so that leads to the next question of how do these relationships and genetics influence response to climate? Is a tree growing in a wet environment better prepared to handle drought? There's some literature that supports that thought. And if that's the case, should we be sourcing these trees for restoration and where we're planting things? Do we want a tree that's then growing in a wet environment because it's more resilient to handling a drought, whereas a tree that's dry is always dry? But a tree that's in a wet environment kind of sees more fluctuations in the availability of water that it's getting. And then how far can we move a tupelo tree for assisted migration? We saw the range there. It's pretty expansive. If we're thinking about migration in response to climate, we're typically seeing a migration farther north latitudinally. And so if we're seeing that, do we want to move a tree that is growing in Texas and just say, hey, now I'm going to throw you in Montreal in Canada? Or is that going to shock it and kill it? Probably. But where's a happy medium? Can we then move a tree from Virginia farther north? And how do genetics influence this movement? And when we're thinking about this movement and like the range of a species, typically those fringe areas of a species range are going to be more genetically limited. And so we could move a tree from New York to Montreal, but that's just going to kind of expand on the genetics that are already most available to move right there. So how can we influence some or incorporate some diversity into that? And then what role can and do cities already play in this movement? And how can they help with forest assisted migration? So I have some processes going on. Very similar to Mia, I'm going to be doing some genetic sampling. I'm really looking at the genetic differences between trees growing in wet versus dry environments, but also so we have the whole range. We have wild versus wild. We have tree growing in Arkansas, Alabama versus tree growing in Virginia, and then we have tree in Virginia growing in wet versus tree in Virginia growing in dry, or dry. And then we also have tree in Virginia growing in a wild setting versus tree in Washington, D.C. growing in a cultivated setting. How are you two related? I want to know. I'm also doing a common garden experiment, and then I'm going to be doing a drought and flood test. And so I'm going to talk a little bit more about the common garden and what that is, because that is really what I am most excited about at the moment. And so a common garden is a planted, genetically diverse monoculture, for lack of better phrasing. You are taking populations from all over the native range of a tree, and you are planting them out in the same location. And when you do this, you are essentially removing all environmental factors that affect growth. So you have, I'll reference this diagram on the right. These are my sample sites. So I went to New York, I went to Virginia, and I went to Alabama, and I did not go to Arkansas. Somebody mailed me seeds. Thank you very much, Stephen. And I have all these seeds, and now they're all going to be grown out in the same location. So we don't see the effect of, a tree from New York growing in a climate in New York. We have a tree from New York now growing out in the same place as Alabama, Arkansas, and Virginia. And so when you see leaf out dates or when you see, growth rates and things like that, that is a direct reflection of something happening genetically as opposed to something happening due to environmental conditions. And so common gardens have traditionally been really relied on and used for economically significant species. So we're thinking the forestry trade, we're thinking lumber, and how can we get the most bang for our buck? And I'm really interested in using this to think about ecologically significant trees and where we can move plants in the context of restoration. And so this past fall, I was on a little seed journey, and I collected over 3,000 seeds across these sites. And now they are winter sowed, so cleaned them all, had a blast. Thank you to my undergrads who helped me. And now we're waiting for them to germinate, and I will have 2 common gardens that will be planted in the spring of 2027 at Blandy Experimental Farm in Boyce, Virginia, and then also at Vassar College up in Poughkeepsie, New York. And so this project or this experiment specifically really aims to provide an understanding of how far can we move these plants. So with the native range kind of keeping bay right in the mid-Hudson Valley, like southern Vermont area, if I'm taking a tree from Alabama to New York and it dies, well, then we shouldn't take that tree and move it to Montreal. But, maybe Alabama and Arkansas are going to croak when they hit New York. But what if Virginia doesn't? And what if Virginia is able to survive? And then what if you can facilitate that movement farther north? We see, we can't move a tree 1,000 miles, but we can move it 500 miles. And that still is good enough to get some genetic diversity in there. Because right now, as I had mentioned earlier, the genetics in New York are going to be pretty limited because it's a fringe. And so if something comes in and starts going after tupelo there, they're probably all very closely related. And so if something's going to get them, they're going to die. And they'll probably all die. And so how can we kind of incorporate diversity into this? And so that's a common garden point. And that's a common garden point. Okay, so now I'm going to digress a little bit and these are photos from fieldwork. It was an awesome fieldwork fall. Got to go to Alabama. Didn't think I would ever be going to Alabama. That's me near a cave. That's a scorpion. Didn't know there were scorpions in Alabama. I got to reconnect with some old colleagues. It was awesome. And so these are just photos of me going out and collecting the seeds. And now I'm going to give you a little test. So it was super fun and awesome. And I love that my job gets to be seeds. It's a great time. But sometimes when you are looking for seeds on a riverbank and they are these little blue berries, you look at the ground and you just see blue rocks. So there are a lot of tupelo berries in here. I'll give you like 5 more seconds to think about what does it look like? Because sometimes it drove me a little insane. And then here are all the tupelo berries. So it's really fun. It's really great. But it can be a bit agonizing at times. And then I'm like, is that a berry? No, it's a rock. Damn. But yeah, so that was a bit of the common garden. The last thing that I just want to touch on while I have you is this kind of like additional thought that I'm thinking about through the context of my research and that I hope to kind of develop more upon. And so this is thinking about cities as conduits for assisted migration. And so cities historically cities and plants have a tricky relationship. Cities have been conduits and introduction points for a lot of invasive species, pests, and disease, predominantly through the commercial plant trade. Could give a whole nother talk on that. But cities are also much warmer and they typically have a pretty distinct climate than that of their neighboring rural areas. And this is predominantly through the urban heat island effect. You're in that concrete jungle, it's very hot. And then people escape to the countryside where there's a breeze and there's a pond and you can actually swim in it. And this warmth can act as a refugia for species who can't quite make it outside of the city yet. And so I think about this in the context of Tupelo, where, I brought up Montreal a few times. That's not an accident. Tupelo are starting to be planted as a street tree in Montreal, which is well outside of their native range at the moment. And that's because they can survive in Montreal right now. I'm really interested in the fact that, this Tupelo is surviving in Montreal. That's great. That's awesome. I'm happy for it. But what if an American robin flies into Montreal, grabs that Tupelo berry, flies 20 miles outside of Montreal, and poops it out? And, right now, probably that Tupelo berry is not going to germinate and that tree is not going to grow. But we are going to hit a climate threshold where that bird, maybe it's a different American robin, is going to eat that berry, poop it out, and that tree is going to take. And then the next tree is going to take. And so when we do our urban plantings in our cities, how can we incorporate assisted migration thought and specifically genetic diversity into these plantings to help kind of shift the framework and the lens in which we view urban environments and natural environments and really have them be more complementary rather than butting heads a bit. And so, some closing thoughts that I want to leave you with is that trees are awesome. I love them. I love all plants, but trees are cool. And they're dynamic and they're constantly evolving organisms. Nevertheless, they and many other plants need our help. Restoration ecology incorporated with forest assisted migration can really hit home on this idea of restoring resiliency. And genetics, which I really kind of skipped over today, but it can encode all sorts of resilience into trees. I'm trying to figure out what they encode into tupelos and why. And whereas, we can figure out what they encode and then we can kind of figure out how that is distributed across the range, then say we need to be prioritizing XYZ area for sourcing these trees. And cities have traditionally been vectors of damage for forests, but we can really change that. And that starts with lobbying for diversity in urban forests. We don't want monocultures. You don't need 800 red maples in Baltimore. You can have 20 and then maybe some tupelos. And a lot of this work is ongoing. I'm still in my first year of my PhD, and this will be going on for the next 4 years. So expect to hear more. I'll be up to a lot of fun stuff this summer. And then I love to end presentations with books because I read a lot. And so these kind of shape how I think about my work. And I've learned a lot from a lot of these authors. And yeah, so these are some good recs. If you're really interested in plants of the South, Saving the Wild South is a great one. And yeah, acknowledgements about the people who make the dream work at the moment. And I'm hoping to add some more folks to this list. But yeah, that's all I got. Should I stop sharing? Should I keep it up?
Hey, thanks so much, Ethan. Great talk.
Thanks.
And if you
Here, I'll
you
stop.
can do whatever you want. Take
I'll
note of
stop.
all those recommended resources. Never fear. We can share those. Yeah, so we can share those resources with you all after the talk. So don't worry about that. And we got some questions coming in on the chat. Lots of good words. Lots of great jobs. Maggie has some questions here. Ethan, are you able to see
Yeah,
her questions in the
I
webinar
can see
chat?
your question. I'm
Okay, great.
ruminating. Um, I haven't given thought, but I am a huge fan of signage. Um, how that signage comes to fruition, I have yet to ponder. But no, I think it's so important, and I think especially from the lens and just like thinking about how we communicate what we do because our work is great and I love all the work that this department and that we all do at Blandy, but it is nothing if it's not communicated to people who it affects. And so I haven't thought about how to interpret that space, but now I'm going to think a lot more. I keep pushing. I'm like, spring 2027. It's so far. It's not far. It's right around the corner.
So I'm going to go ahead and stop the live stream just because